Here we get ready to discuss what may be the most important topic in AMP1, the concept of membrane transport and its different forms that it will take on. Most of what we will talk about moving forward, whether it be neurophysiology, cardiophysiology, renal physiology, respiratory physiology, will have a very high percentage of membrane transport as part of its topic. Of course, here we're going to discuss it in its generic forms. We're going to discuss the different types, what drives it, and what sorts of solutes move back and forth the membrane in such ways. And then we're simply going to take those concepts and apply them to particular systems. It isn't unusual for certain topics in AMP2 that I cover that I also post this particular lecture for those students so they can refresh on membrane transport because it is such a foundational topic. We have to understand the terminology. First and foremost, you have to know the language that we're going to use to keep things straight. And simply put, we're going to go through the various types of membrane transport and understand the nuances that are associated with it. Now, each type of transport is going to come with a definition per se. I would encourage you not to simply memorize the definitions of the types of transports, but rather why we need several different types of transport. Each type of transport will become a player in the game, so to speak, when the characteristics of the solutes become different. They all have different applications. They all accommodate different types of solutes. So it's important to realize why we need different types of transport rather than the definitions of those types of transports to start with. So first off, the basics, solute versus solvent. A solution implies we have more than one item in the same beaker. Trying to keep this as simple as we can. A mixture, another chemical term, would imply that we have two different items mixed together with the high probability of them staying separate once they're mixed. Think about clay put into water. That will eventually precipitate out, and you'll be left with, for the most part, clear water and this clay settled to the bottom. A solution implies we have the two items mixed together in much more of a homogeneous fashion. Salt added into water. Yes, of course, we could add enough salt so that eventually no more will dissolve. But when you have salt water, you have those sodium chloride ions equally dispersed through that watery solution. In a situation like salt water, the water is the solvent. It's the liquid portion. The sodium and chloride ions then become the solute. Certain types of transport we're going to discuss, well, most types, will involve movement of the solute as it moves from the watery environment of the intracellular fluid to the watery environment of the extracellular fluid, or vice versa. But when we talk about osmosis, it's important to realize that really what we're talking about is the movement of the solvent as it moves back and forth. Remember that any time we're talking about membrane transport, we are talking about the ability of a solute to move back and forth the membrane, entering the cell or leaving the cell. So this is the mass that we're trying to traverse when we're talking about solute or solvent movement. Through the phospholipid heads, through this very strong hydrophobic environment that we got into when we discussed the membrane structure, and then out the other side. So if we're a solute and we're trying to make our way through this membrane, we have to be able to make our way through the phosphate heads, be at home in this non-polar hydrophobic environment to get through, and then out the other side. And again, we could talk about different types of fluid compartments, and of course we will as we go through AMP 1 and 2. But for now, we're talking about movement into and out of the cell. Now later on, we may apply some of the concepts of this movement into and out of a capillary. But that's not what we're discussing now. It's purely across a cellular membrane into and or out of a cell. We used the analogy of ping pong balls floating on water when we discussed the phospholipid bilayer. If we were looking down at this membrane, this is what we would see. These round ping pong ball-esque phosphate heads, and because they're spherical in nature, they don't pack together, so there's going to be small openings in between, giving us the opportunity of a solute to be able to pass through there. However, remember, as soon as we pass through that phosphate layer, we are in a very strong hydrophobic environment. So if a molecule is too large to pass through and fit between the phosphate heads, it's going to get rejected. But likewise, even if a molecule or ion can fit through that phosphate layer, the small openings, if it is going to be repelled by the very strong hydrophobic environment deep in the membrane, then it's also going to be not permitted to pass through. It's going to be repelled by that environment inside the membrane. Remember, we're dealing with a fluid mosaic model, so we're trying to reintroduce some terms that we had from our membrane discussion. Some of the proteins that we have embedded in the membrane are these tube-like structures to aid in passage across the membrane. So for items that maybe can't pass directly through, we may have passageways embedded in the membrane to accommodate. Of course, those can be drawn or illustrated in a number of different ways. Simple protein tubes like the one on the far left, to more of a Pac-Man looking shape as a solute binds to it, and then the protein changes shape and then kicks that solute out the other side. So here is our different types of transport. Two broad headings, passive versus active. That's really where we're going to start to differentiate. We're going to break this down and compare and contrast different types as we're able to partner them up, so to speak. So active versus passive transport is where we're going to begin our differentiation. From there, we will look at the different types of passive transport, diffusion, and then simple versus facilitated diffusion, on why we need two different types. Then we'll look at our next type of passive transport, being osmosis. And then lastly, we'll finish off with our two different types of active transport, primary versus secondary. Now, already, even though we are going to compartmentalize each of these and kind of knock them off somewhat one by one, it's important to realize that by the time we look at a cell and a functioning cell, all of these different types of transport have the opportunity to be ongoing in a cell at the same time, somewhat in a synchrony. So active versus passive transport. First of all, there are two main differences that you are going to need to understand. What drives the transport is the first one. Now, when I typically ask this question in class, what drives active transport? People will say energy or ATP. And then when we start talking about passive transport, they will start giving definitions such as, oh, that's transport that doesn't require energy. Now, already I want to get us out of the habit of this. We spent some time in one of our first chapters talking about energetics. Remember, energy doesn't just manifest. What we see are types of energy moving and transforming from one form to another. And energy in the form of ATP is not the only form of energy that we discussed. We can have energy in the form of a concentration gradient. A concentration gradient is when we have a lot of one solute on one side of a membrane and very little of that solute on the other side. So when it comes to active transport, it relies on energy in the form of ATP. Passive transport, however, will rely on energy as well, but this time in the form of a concentration gradient. As long as we've got ATP present, we can get active transport. As long as we've got a concentration gradient present for a particular solute, then we can get passive transport of that solute. The second main difference is the end result. If we let each type of transport occur undisturbed for a period of time, what's our end result? For active transport, it's going to be the concentration gradient production. We're going to create a concentration gradient. We are going to use ATP to essentially pump solutes across the membrane and accumulate them on one side. So again, from an energetic standpoint, look what we've done. We've used ATP to drive active transport and create a concentration gradient. So we've transformed energy in the form of ATP into energy in the form of a concentration gradient. We've pumped them across. Passive transport, however, if we let continue, the end result is going to be what we call an equilibrium. As we see these solutes passively transport across a membrane, we will start to use up our concentration gradient. At the time when we have equal concentrations on each side of the membrane, we no longer have a concentration gradient that we can use to power that passive transport. And at that point, equal concentrations on each side of the membrane, we call that an equilibrium. So those are our two main differences between active and passive. So now let's talk about passive transport. Where we need to start first is just simply talking about molecular movement. Let's talk about general movement of molecules to begin with, and then we'll apply it to the cell. It becomes so much easier. Passive transport will always involve random movements of molecules, no matter what matter we happen to be talking about. The atoms in that matter are undergoing a certain degree of movement. If it's a glass of water, the water molecules in that glass of water are constantly in motion throughout that liquid, bumping off of each other as they go. The salt water solution that we talked about earlier, now you've got the water molecules bumping around in there, and you've got sodium and chloride ions also continuously bumping around in there. Even a solid, even though they won't be migrating through that mass, there'll still be a vibration. So the movement of these solutes through a solution, if it's passive, is always going to be random. And when we say it moves from a concentration gradient, what we're really talking about is the likelihood. The random movement of those molecules will always cause a general movement in the direction from a high concentration to a low. But the movement is always random. When we look at this random movement, you will come up with a topic in the lab called Brownian motion. So when we watch this Brownian motion occur, we're going to see random movement before our eyes. We're seeing a styrofoam ball placed in a small little dish of brass beads. We vibrate the brass beads just like you would see with the molecules bouncing around inside a solution. And as they collide, we're watching that styrofoam bead get jostled around in a random manner.
Maybe it'll go another way. And it does. And each time you do it, you might get a completely
Now, I put this video in an isolated form in class. You can watch just this video alone. But if we were to use that as a model and even represent concentration gradient with it, if we took 6 of those little styrofoam white beads and put them in one place in that dish and then vibrated the whole system, you would see those white styrofoam beads over time, randomly, through random movement, spread out into an equal concentration across the top of that dish. Now, does that mean that at any given time, some are going left, some are going right? Absolutely. It is random. But that is going to be the premise of any passive transport. It's the random movement that will always ensure that those solutes move from a high concentration to a low. And that'll continue until we can no longer get any net movement. We discussed the concept of equilibrium earlier. Now, when diffusion reaches equilibrium, that doesn't mean the movement stops. It simply means we lose the likelihood of movement in any one general direction. Once we've achieved equilibrium, the random motion that created that equilibrium still continues. But now the net direction. There will be no predictable movement of solutes in any one direction once we reach that equilibrium. And when we say net movement, at that point, if we're talking about movement into and out of a cell, even though there might still be molecular movement crossing that membrane, there's no net gain in one direction versus the other. And the concentration gradient at that point is deemed exhausted. So here's what we would really look at if it was a diagram looking scenario. Picture this as being Kool-Aid. On the left, we have all of our concentrated Kool-Aid crystals. We just dump those crystals into the vat of water and we leave them be. Over time, we will start to see it move from this very high concentrated solution in one portion of the beaker to a nice, even, homogeneous movement. Random movement of all these solutes bouncing off of each other as they collide through a solution will cause them all to generally move from a high concentration to a low. They'll move from this corner of the beaker and spread out. Once they're all spread out, like the vat on the right, this is equilibrium. This is a pure homogeneous solution. Yet you're still going to see these red solutes move in random directions. One will move up, one will move down, one will move left, one will move right, but because of the random movement now, there is no net movement in any one direction. And you'll see that in the lab as well. You'll do potassium permanganate in beakers of water. And you can see how on the left we start off with a nice dense concentration in one part. And through random movement, you will see that color spread out throughout the vat of water. Now, you're witnessing hot water on the left and cold water on the right. We will make a point of that later on in this presentation. But regardless, we're talking about random movement. So you can still see some darkening areas in that vat on the left. You come back after a half hour and it will be a nice consistent color through the random movement. Once we reach that point, that is when we no longer have a concentration gradient, but those potassium permanganate solutes in the glass of water are still moving about inside. So we start with a high concentration on one side of the membrane, as we see on the left of this diagram. Because it's randomly moving, if we put a membrane in between here, like we were going to see with movement into and out of a cell, we put a membrane in between, random movement from a high concentration to a low will dictate that some of those will move across the membrane to the bottom of the screen. Of course, they can't move in the other direction initially because there is none over there. But we see these three make their way across. Some more time goes by. We see a couple more make their way across. Now, these first three in the middle section that finally made their way across, random movement still means that they may go back across in the other direction. But because we've got more on one side than the other, we're always going to be more likely to get random movement in the direction from a high concentration to a low. Now on the right is when we have our equilibrium. Some of these on the outside may move in, some of these on the inside may move out, but at that point there was no net gain. One enters, one leaves. We still saw diffusion, but not in a manner that caused net transport. Now we've got an equilibrium with random movement of solutes throughout that solution across the membrane. Now, what we'll see in the cell very quickly is that we'll never just have one solute involved. Already we've talked about some membrane potential stuff, and we've had at least 5 different ions involved in it. Whenever we're talking about diffusion, every single individual solute behaves as if the others weren't present. So here we've got a situation where we've got a membrane down the middle. We've got the left chamber with all of the red solutes, and on the right we got the white solutes. As we let that passively diffuse across that membrane, we will see the reds move from left to right in the direction of their concentration gradient, as if the white ones weren't even present. And we will see the white ones move from right to left along their concentration gradient, as if the red ones weren't present. They will both go from a concentration gradient until they reach equilibrium, as if they were the only player in the game. Movement of the reds don't affect the whites. Movement of the whites don't affect the reds, period. They will each move independently based on their own concentration gradient. So how quickly we see solutes diffuse will depend on a number of different variables. We're going to knock these off the list here first, but I do want to point out right away: solute characteristics, surface area available for transport, and the strength of the concentration gradient. Those 3, we cannot talk about any one of those individually without incorporating the other 2. Those 3 variables are very intimately related. So hopefully by the time we get to the last one, you will see exactly why we can't discuss one fully without at least introducing the other 2. So first up, temperature. Simply put, the higher the temperature, the faster the rate of diffusion. We said diffusion acts as a random movement of molecules as they collide off of each other in the solution. Well, the higher the temperature, the faster these molecules move. In a solid, you heat it up, they vibrate quicker. A warm glass of salt water has those sodium chloride ions moving through that solution quicker than a cold glass. And you will see that in the lab. You'll put your potassium permanganate in a cold vat of water, you put some in a hot vat of water, and you will leave them be. What you'll see in the lab is that in the warm vat of water, it'll reach equilibrium in about a half hour, maybe quicker, depending on how hot the water was. The cold vat, the room temperature vat, your lab session will be over, and it will still look like a solution that has light and dark areas in it. The higher the temperature, the faster the rate of diffusion. The second variable was distance traveled. Now, in this situation, where we had vats of water, obviously a 200 ml beaker of water will reach equilibrium quicker than a 50-gallon drum, because those solutes now have greater distance that they have to carry in order to reach equilibrium. But we want to make sure we're applying this to the cell as early and as often as possible. So when we're talking about distance travel, we're really talking about the thickness of the membrane. Now, in most situations, the cells that we're going to be discussing, the membrane thickness is not going to vary much significantly. So we can consider this to be somewhat of a constant. So because of these 2 variables, temperature and distance transport, from a cellular perspective, we can disregard them. We are not going to see variations in temperature to such a degree in the human body that we're going to see them dictate diffusion rates. Likewise, as long as we're talking about diffusion into and out of a cell, we can negate distance transported, because pretty much every cell is going to have roughly the same plasma membrane thickness. This concept will become important later on when we do cardio, but as far as diffusion simply into and out of a cell, temperature is not a variable we're going to consider, neither is distance transported. So that leaves us with our last three, our solute characteristics, our surface area available, and the strength of the concentration gradients. Let's talk about those. The solute characteristics. The larger the molecule, the slower it moves. Small solutes move through solutions faster than large solutes. So if we were talking about movement into and out of a cell, we would expect those small solutes to traverse the thickness of the membrane quicker than a large one. Okay, we're going to come back to that. But remember, large solutes that we talked about cannot simply go through the membrane. They need help. So yes, if it was just a beaker of water, small solutes move through that beaker quicker than big ones, but we're talking about a cell here now. If we are looking for that solute to diffuse across the membrane, they need to be small enough to fit through the openings in the phosphate layer. So that introduces us to our two different types of diffusion, simple and facilitated. Now let's look at the definitions first, although I said I didn't want us to learn this based on the definitions. Simple diffusion is diffusion through the membrane, directly through the membrane along its concentration gradient. It goes between the phosphate molecules and traverses the membrane directly. Now, because the openings in between those phospholipid molecules are small, this is really only going to apply to small solutes. If all we're looking at is size. Facilitated diffusion, as the name implies, to facilitate means to help. They utilize protein channels to help solutes that otherwise couldn't get across by simple to make their way through that membrane. Now, based on size alone, this is going to open up larger openings for those larger solutes. So if this glucose molecule couldn't fit between the openings in the phospholipid molecules, we put a protein channel in here to accommodate its size. And again, you'll see those drawn in several different mechanisms. Simple protein tubes, these Pac-Man looking shapes. When we get to neuro, you'll look at voltage gated channels and you often see them drawn like this doorway system. Next up is the polarity or the charge. Is it an ion that carries a charge versus an atom? Is it a polar covalent molecule versus a nonpolar? This comes down to what sort of environment these solutes are most at home in. Remember, the inside of this membrane is very strongly hydrophobic, water hating. So if you are a hydrophilic solute, like an ion, even though you're small enough to fit in between the phospholipid molecules, you are not going to be able to pass through that highly hydrophobic environment. Now, some people might say, well, what if it was just moving really fast and it could just blast through there like a dog with a shock collar? If it builds up some momentum, it'll be through the fence before it even gets shocked. Think about the scale we're working on here. We're working on a molecular scale. This sodium ion diffusing directly through this membrane when it's very hydrophilic and the inner membrane is very hydrophobic would be like looking at a football field. You would be in one end zone trying to make your way across the rest of the field, while every player you could muster was on that field trying to knock you backwards. These sodium ions, or any ions for that matter, even though they can fit through the phospholipids, they are not going to be at home in the hydrophobic environment. Therefore, they're going to be repelled. So how do we accommodate? Facilitated diffusion. Not only can those protein channels accommodate the larger molecules, but they can also accommodate the very hydrophilic, polar, or charged ions. So now we read off the definitions earlier, but I didn't want you to learn these based on the definitions. I want you to realize why we need two different types. We have two different types of diffusion because we've got a range of solutes that may need to diffuse across the membrane. So if you are small and hydrophobic, nonpolar, uncharged, you will diffuse across that membrane by way of simple diffusion. You have to check both of those boxes, small and hydrophobic. You can fit between the phospholipid heads, and you're comfortable in the fatty acid environment. You have to check both of those boxes to utilize simple diffusion. For facilitated, any solutes that could not utilize simple diffusion gets help by these facilitator proteins and will diffuse across the membrane using facilitated diffusion. So that was our solute characteristics. Now already, if we look at the second to last variable on the list, surface area available, hopefully you're already realizing why these are integrally related. Because if we look at simple diffusion, the surface area available is, for all intents and purposes, the size of the cell. Small cell over here on the left has a much smaller surface area than the large cell on the right. And because we're just talking about movement through the membrane directly, the entire surface of the membrane becomes available for transport. When we're talking about facilitated diffusion, though, the only points of entry and exit are going to be those protein carriers. So look at this scenario. We have a large cell on the left with 2 protein carriers. However, we have a smaller cell with 4. In this case, the smaller cell, with respect to facilitated diffusion, actually has the greatest surface area available for transport. So we can't really talk about surface area available without looking at the differences between simple and facilitated. And the reason we have those two different types is because we can have different characteristics of solutes. Large ones, small ones, hydrophobic, hydrophilic, polar, nonpolar, charged, uncharged. The characteristic of the solute dictates the type of transport. And the type of transport basically has different contexts by what we mean by surface area available. The next variable that becomes intricately related with the previous two is the strength of the concentration gradient. Now here is our general rule. The stronger the concentration gradient, the higher the rate of transport. The stronger the concentration gradient, the higher the rate of diffusion. I guess I should say more specifically, not just transport. We're talking about diffusion. On the left, we've got 100 milligrams per liter of a solute on the outside of the cell. We've got 10 milligrams per liter intracellularly. That is a concentration gradient. We got more on one side of the membrane than the other. We will see solutes move from the extracellular fluid into the intracellular fluid. On the right, we've also got a concentration gradient, but it's not considered as strong. The difference between those two compartments of fluid is not as dramatic. Therefore, we will see in this snapshot, the scenario on the left will have the higher rate. Now realize that we are moving towards equilibrium whenever we're witnessing diffusion occur. So we started off with the concentration gradient on the left with its rate. But if we let that diffusion occur over a period of time, eventually we're going to see it get closer to equilibrium. And then we come back a little bit later, and it might be 55-45, closer to equilibrium again. The closer we get to equilibrium, the slower the rate becomes, because now we may have an increased likelihood of those solutes going back in the opposite direction, negating any transport in the direction with the concentration gradient was dictating. So the stronger the concentration gradient, the higher the rate of transport. Now, for simple diffusion, that is an absolute rule. For simple diffusion, it is 100% true. The higher the concentration gradient, the higher the rate of transport. However, once we look at facilitated diffusion, we've got a problem. That is not 100% true for facilitated diffusion, because the protein transporters themselves become a limiting step. In a classroom, I always use an example of the fire alarm going off suddenly. And picture you're in a classroom with 30 students. The concentration of students is 30 inside the room and none outside the room. So we've got a strong concentration gradient of students intracellularly. If you were able to pass through the walls like we would see with simple diffusion, picturing the walls of the room is the membrane. If you could pass directly through the walls and that fire alarm went off, we could clear that room in about 3 seconds. The rate is going to be high. However, if you were limited to only using the doors, like the analogy implies with facilitated diffusion, all 30 of you would rush towards the door and you would bottleneck, because that door can only allow 1, maybe 2 of you to leave at any one time. So now that rule of the higher the concentration gradient, or the stronger the concentration gradient, the higher the rate of diffusion, is only true up to a point for facilitated diffusion. After that point, if we pile on and we create an even stronger concentration gradient, that will not result in a higher rate of diffusion to take place. We've exceeded the ability of the protein carrier. Now, practically speaking, you will see that come up, especially in renal. When you look at the function of the kidney, you will see the concept of a transport maximum, the maximum rate at which any protein carrier can operate. And this is really how your kidney reestablishes blood concentration levels. Think about somebody with diabetes. If you're a normal person, you don't see glucose in the urine. But if you're a diabetic and your blood sugar is 3 times that of normal, you certainly do. Because as a normal non-diabetic, you filter all the blood and you've got enough of these protein carriers to reabsorb all that glucose. But if you are bombarding those carriers with 3 times the amount of glucose, they simply don't have the time, they don't have the capacity to reabsorb all those glucose molecules in the given time that they have. Therefore, any excess glucose spills over into the urine. So, simple versus facilitated. They both require a concentration gradient. You will always see random movement of solutes in the direction of high concentration to a low. For the simple diffusion, the higher the concentration gradient, the higher the rate of diffusion. For facilitated, that's only going to be true up to a point. Every protein carrier has a maximum rate at which they can operate. And for simple diffusion, if a solute is going to utilize it, it has to check 2 boxes. It's got to be small, and it's got to be nonpolar or uncharged. If we don't check both of those boxes, then it automatically becomes a situation for facilitated diffusion. So at this time, it's important to go back and reintroduce something that we mentioned earlier. We are going to have numerous solutes present. If we only have one solute, it will diffuse across based on its own, in a mechanism based on its own characteristic, from a high concentration to a low. If there are 100 different solutes involved, they all still only behave based on their own individual characteristics and concentration gradient. The presence of any other solute doesn't influence the diffusion or its rate at all. They are completely independent events. So here, we got 4 potassium ions inside the cell. We've got 4 sodium ions outside the cell. 4 inside, 4 outside. But because they're different, this is not an equilibrium. Sodium will diffuse in as if the potassiums weren't there. Potassium will diffuse out as if the sodiums weren't there. Independent movement based on their individual concentration gradients. They will continue to move across the membrane until they reach equilibrium. So there'll be 2 potassiums inside, 2 potassiums outside, 2 sodiums inside, 2 sodiums outside. Once we reach that equilibrium, they will still be able to randomly move across the membrane, but they will very likely trade. 1 sodium will leave, 1 will enter, at which point that is considered 0 net transport. Now, if we have solutes present that cannot diffuse across the membrane, then we open up the opportunity to have a different type of transport called osmosis. In order for osmosis to occur, you have to have a solute present that is unable to cross the membrane. So it's got to be a semi-permeable membrane. In which case, the solvent, the liquid, in our case it's always going to be water, the solvent will move in the direction of the high solute concentration. So to go back to a drawing that we have here handy, let's assume for a moment that those sodiums can't diffuse across. They're stuck out here. Now water will leave the cell and go out into the high concentration of sodium. So it's solvent movement. Now this is going to be dependent on the total solute concentration, not any one particular solute. So these could be 4 different sodium ions, or they could be a sodium, a calcium, a magnesium, and a phosphorus. If all 4 can't cross, all 4 will exert the exact same osmotic influence as if they were all the same type of solute. It's a particle count, not a measure of any one particular solute. And because of that, we try and stay away from terms like concentration. Concentration implies we're talking about a very particular solute, like sodium, like potassium, like calcium. The term osmolarity will take the sum of all of those into account. And the presence of any solute that cannot cross that membrane will exert an osmotic pressure on the solvent on the other side. Here we see a horseshoe flask with that semi-permeable membrane here in between, represented in green. We put the solute in one side. The solute cannot cross that membrane. The nature of this membrane prevents the solute from crossing. We will see those solutes, those pink solutes, exert an osmotic pressure. And we will see solvent move across the membrane in the direction of the high solute concentration. Driven by that osmotic pressure. And we can measure what that pressure is if we take a plunger and stick it down the left-hand side here and see how much force it takes to get the water level to even out again. We describe concentrations on one side of a membrane with compared to the other with three different terms: isotonic, hypotonic, hypertonic. Let's look at the hypertonic first. Hyper as a prefix typically means above, more than. So if we have a membrane with a high concentration on one side, we consider that side to be the hypertonic side. This is the hypertonic solution. Hypo as a prefix generally means below or lesser than. So conversely, if this is the hypertonic side on the left, on the right-hand side of that horseshoe solution, horseshoe flask, is the hypotonic side. It's the side with the lower solute concentration. Isotonic would be a term used to refer to both sides of the membrane having equal concentrations. Solvent will always move in the direction of the hypertonic side. Now, in the lab, you'll be referring to hypertonic solutions and hypotonic solutions in the same way we looked at this scenario. One membrane, this is the hypertonic side, this is the hypotonic side. But it's important to realize how we phrase this from a cellular perspective. You will get questions along the lines of, a red blood cell is placed in a hypertonic environment. That implies that the extracellular fluid is the hypertonic side. We usually use these terms when we're describing cells to describe the extracellular fluid. A red blood cell is put in a hypotonic environment. A red blood cell is put in a hypertonic environment. We're using these terms to describe the extracellular fluid. Here we see red blood cells. Here in the middle, normal, isotonic. Isotonic means we have the same concentration on both sides. Solvent moves in the direction of the high solute concentration. In this case, we don't have a high side. We don't have a hypertonic side. So we get no shift of fluid by osmosis. But if we put a cell in a hypertonic environment, we will start to see water leave the cell, as shown as we move to the right of this diagram. We're starting to see water leave these cells. The cells are shrinking up. They're losing mass. They're losing water. And of course, that continues for too long, and these cells are going to be dead. If we put a red blood cell in a hypotonic environment, now water will move into the cell because that's where the high concentration is. These cells start to plump up. Some of them can take on so much water, and the membrane becomes stretched so thin that they can barely even be seen on the slide anymore, called a red blood cell ghost. Now, we said losing too much water was detrimental to the cell as it becomes crenated. What do you suppose happens if the cell takes on too much water? Not much different than if you put too much air in a balloon. It's going to burst. We call that a lysis. When we look at this from a feedback mechanism, we haven't introduced many feedback mechanisms specifically yet, but this is one you will talk about with regard to the kidney and the blood in AMP2. Blood volume regulation. Think about what happens when you lose too much fluid from the body. You become dehydrated. Chemoreceptors, measuring blood osmolarity, start to detect an increase in concentration. You lose fluid, but what you're left behind with is still all the proteins, all the cells, all the electrolytes. Receptors detect this change in concentration. And it's going to perceive that as a loss of fluid. The control center interprets what the receptors are telling it, and you will get organs recruited to counter that change. Remember, negative feedback. In this case, the kidney will stop producing urine. Or if it does, it's going to do so in a manner that preserves as much water as possible. You're going to produce low volumes of very concentrated urine. The mucous membranes in the back of the throat are going to dry out as saliva production goes down. It's going to give you this sensation like you want to consume fluids, drink water to get that fluid volume brought back up. The last type of transport we have to get into is active. Now, we said this requires the use of ATP. This is a pump. We're trading ATP, energy in the form of ATP, to produce a concentration gradient. Energy in the form of a concentration gradient. We have two different types: active, primary active, and secondary active. Now, in class, whenever I ask people what's active transport, remember I said I always ask that question early on, and they say uses ATP. What they're describing, without even really probably realizing it, is what they're describing is active primary transport. Uses ATP. The ATP is broken down directly at the site of transport. So the protein channel that the solute goes through, that's where the ATP is broken down. And it will do so, as long as we have ATP, we'll pump those solutes across, accumulating them on one side, creating a concentration gradient. Secondary active transport is still considered active transport because it has to meet two criteria. It's ATP dependent, and it creates a concentration gradient. If we check both of those boxes, it's active transport. But we can have a scenario where we don't get ATP broken down at the site of transport. Now, for this, we are not so far into this that we can't use simple comparisons. I would like you to think for now that secondary active transport is actually driven by the efforts of the primary. So we use primary transport to break down the ATP, create a concentration gradient. Then we use the energy in the form of that concentration gradient to drive the secondary active transport. And hopefully when you look at some diagrams, that'll become clear. Secondary active transport always moves at least two solutes. It'll either move them in the same direction, call it a symporter, or we'll move them in opposite directions, and we call those antiporters. But they always move at least two. Forget that diagram on the left of a uniporter. Now, some of the active transporters that we're going to be discussing, well, the first one on the list you may get sick of hearing about, the sodium-potassium pump. We'll be talking about it in every system. It's a major contributor to the production of our membrane transport that comes up early. Every ATP we break down, we pump out 3 sodium ions, we pump in 2 potassium ions. We have our hydrogen potassium pump in the stomach as part of maintaining a highly acidic stomach contents. In muscle, we have our calcium pump. We have a bunch of calcium sequestered in the endoplasmic reticulum of a muscle cell. In order for you to be able to contract that muscle, that calcium has to be released. Once you're done contracting, you pump it back into that sarcoplasmic reticulum. Now, if we want to look at how this could occur in a practical situation, remember we already discussed ATP production, let's look at what happens in a situation of a heart attack or a myocardial infarction. We get a blockage of blood. That prevents blood from reaching the heart tissue, decreases the oxygen that it needs. As we saw with our oxidative phosphorylation, if we don't have oxygen present, we have a hard time producing ATP. ATP goes down, we no longer have the energy we need to use our sodium-potassium pump. We can't pump sodium out, therefore now it starts to accumulate in the cell. Becomes toxic in the cytosol. The osmolarity of the cytosol goes up, water moves into the cell and causes that cell to burst and lyse. So here is on the left. Let's look at this scenario first on how we can use the efforts of the primary to drive the secondary. The red transporter on the left is your primary active transporter, your sodium-potassium pump. You're using ATP, breaking it down, pumping sodium out and potassium in. And we're going to create a concentration gradient of sodium on the outside and a concentration gradient of potassium on the inside. Now, it's at this point where we have to understand, like I said earlier, that all these different types of transport occur in a harmony. Because look at what just happened. We used ATP to pump sodium out. We created a concentration gradient. What transport did we already talk about that used a concentration gradient? Diffusion. So the stronger of a concentration gradient we create by pumping sodium out, the stronger of a driving force we've created for that same sodium to diffuse back across the membrane. Now, if that sodium is going to naturally want to diffuse back across the membrane anyway, we may as well take advantage of that. Here, in orange, we have a secondary active transporter, allowing that sodium to come back in. But as it does, it makes sure that it kicks a calcium out. So now, this being a secondary active transporter in orange, do we check both boxes for active transport? We're creating a concentration gradient of calcium, and yes, it's ATP dependent, but back here. Because if we don't have ATP present, we don't get our sodium concentration gradient. Without our sodium concentration gradient, we can't have sodium entering the cell back here again. And it's all based on the premise that every time we create a concentration gradient, we increase the driving force for diffusion in the opposite direction. This is really like a leaky rowboat. You have to keep bailing it out. You will need to maintain these active transporters constantly to break down ATP, so we can keep concentrations of these ions on one side of the cell versus the other. And by using a major primary, like our sodium-potassium pump, we can use that sodium concentration now as a bit of a universal currency, because we can have a symporter with sodium and chloride to pump chloride in, sodium and amino acids to pump amino acids in, sodium and glucose to pump glucose in. We can drive any number of secondary active transporters based on the efforts of the primary. You'll see that practically when we do digestion. On the right-hand side of this diagram is the food you just swallowed. On the left-hand side is the blood. You can have a primary active transporter here at the basement membrane, pumping sodium out, potassium in, another sodium-potassium pump, creating a low concentration of sodium intracellularly and a high concentration extracellularly. But now, we've got the salt you just ate diffusing into the cell from a high concentration to a low, but now dragging the glucose with it. Without the sodium-potassium pump, we would not have created the concentration gradient across this apical membrane. Therefore, we would not be able to actively transport glucose across. So sometimes we don't just use it along the same membrane, but we can use it in a tissue layer, primary on one side of the cell, the secondary is on the other, and we can start actively transporting any number of solutes that you just ate into the blood. Other types worth talking about, although they won't really be notable until we get into immune, is bulk transport. Now we're not talking about things at the molecular level. We're talking about large volumes of fluid. Endocytosis versus exocytosis. Bulk movement into and out of a cell. Penocytosis is bulk movement if we only have a liquid. What you might have heard about is phagocytosis, when you have a white blood cell engulfing a bacteria. Now we can have receptor-mediated endocytosis. Starts over here on the right side of the diagram. You got receptors in the cell membrane attracting a solute. When they do, this part of the membrane connects with the opposing portion, and it brings that entire vesicle into the cell. We merge that with lysosomes, digest what you've brought in, and then we can take those receptors again and recycle them to the external surface of the cell once again. From an immune function, you see white blood cells form these pseudopods, wrap around the bacteria and completely engulf it. Now, once it's in here, you can use the digestive enzymes inside lysosomes to break it down further. See, this white blood cell kind of, quote unquote, chasing this bacteria. It's not really chasing it. That bacteria is just randomly moving based on thermal energy. But you'll see at one point it finally catches up to it, and it just wraps itself around it. Right here and gone. So it just used its own membrane to engulf that bacteria and bring it into the cell where it can now be digested. So get your terminology down. Solute, solvent, concentrations, osmolarity, isotonic, hypertonic, hypotonic. Get your terms down so you're not misinterpreting something with the language. Then understand that we can have all of these different types of transport based on, one, the nature of the solute, and two, what we want to accomplish. And then once you get the individual types down, step back from it and understand that we can have all of these different types of transport occurring at the same time. We can use ATP to actively transport a solute out, but it's going to want to diffuse back in at that point. So we got to keep pumping it out to maintain the concentration gradient we're trying to create. Any solutes that we have present that can't get across, we're going to see osmosis shifting the solvent back and forth. So this is one of the most important topics that you're going to learn in A&P 1. And if you don't understand the different types of transport, it's going to haunt you with every single system that we come across. So any questions you have about this, try and reach out quickly, please, so we can get them taken care of as soon as possible. Otherwise, by the time we get into neuro and you realize that you struggle with this, it will already be somewhat too late. So good luck. Reach out with questions, and we'll be trying to touch on this subject in many different ways.